Hello students, today we will be learning Chapter 12 Respiration in Plants Theory Part 3. This will cover NCERT sections 12.5 to 12.7. This is the disclaimer. Now let's look at the learning objectives of our today's class. So we will understand the respiratory balance sheet, which shows input and output of compounds during respiration. Then we will explain amphibolic pathways' dual role both in catabolism and anabolism. Then we will define the respiratory quotient, which is RQ, and we will also calculate RQ values for different substrates. So we will also interpret the RQ values to identify the respiratory substances in plants. So now let's look at our index. So we will study the respiratory balance sheet, which is NCERT section 12.5. Then we will move to the amphibolic pathway, which is NCERT section 12.6. Then we will discuss the respiratory quotient, which is NCERT section 12.7. Students, then we will look at the summary section where we will take a small recap of what we will be learning in today's session. And then we will finally solve some NCERT based questions. Then we will look at some keynotes. We will highlight certain important points. And finally, we will look at some interesting questions which will be for you to solve. So let's begin with the respiratory balance sheet. So let's see how plants produce energy through respiration by calculating the net ATP yield. So let's calculate the net ATP yield from one molecule of glucose, which is a theoretical calculation. But before we talk about numbers, let's discuss some key assumptions. So these assumptions, they simplify the process for calculation purposes, but they don't always reflect reality. So the first assumption is, we assume that the pathway is sequential and orderly, that is with no side reactions. However, in reality, cellular processes, the reactions are more simultaneous. The second assumption is, we assume that NADH from glycolysis is transferred directly into the mitochondria. However, in reality, this transfer can be less efficient, affecting the total ATP yield. Our third assumption is, so we assume that no pathway intermediates are used for other synthesis reactions. In a living cell, however, intermediates might be diverted for other purposes, which may impact the energy production. And finally, we assume that only glucose is the substrate. But in reality, plants, they do use other substrates like fats and proteins, which can alter the ATP yield. So these assumptions are only for us to simplify the calculation. But always remember the complexity of real life, that is real cellular processes. So let's look at the scenario in real living systems. So all the pathways, they generally operate simultaneously if you look at real cellular processes. Because the efficiency of these processes, it can be influenced by various factors, including environmental conditions and the plant's metabolic state. Now substrates, they flow in and out as needed. And in the process, ATP is used continuously. And another thing is that enzyme activity has multiple control mechanisms. So let's look at the ATP calculation. So here in the table, you can see the pathway. So first, let's talk about glycolysis. So from glycolysis, we get 2 ATP. That is the direct ATP, which is substrate level. 2 NADH gives 6 ATP. So in glycolysis, there is no ATP from FADH2. There is ATP from NADH. So the total ATP produced in glycolysis is 8 ATP. If we talk about the link reaction, that is oxidative decarboxylation, there is no direct ATP from the process. From NADH, from 2 NADH, we get 6 ATP here, and therefore the total ATP is 6 ATP. But from FADH2, there is no ATP production. That is, there is no contribution from FADH2 in ATP production. So we saw for glycolysis and link reaction. Now if we look at Krebs cycle. From Krebs cycle, if you remember, we get 2 ATP as GTP. Contribution from NADH is from 6 NADH, we get 18 ATP. And if we talk about the contribution of FADH2, from 2 FADH2, we get 4 ATP. So the total ATP production is 24 ATP. So if we look at the total count, direct ATP from all these processes is 4 ATP. From NADH, that is total from 10 NADH, we are getting 30 ATP. If we talk about FADH2, from total 2 FADH2, we are getting 4 ATP. So if you plus all this, you get total ATP to be 38 ATP. Now let's make a comparison between fermentation and aerobic respiration. So first, if we look at the definition, fermentation is an anaerobic breakdown of glucose to produce energy. So much less energy is produced in fermentation, we have seen, as compared to aerobic respiration. It is just the 2 ATP molecules we get from glycolysis. If we talk about aerobic respiration, it is the complete breakdown of glucose in the presence of oxygen. So there is complete oxidation of glucose that is happening here. And the major energy that is produced from aerobic respiration. Now, another major difference, if we look between fermentation and aerobic respiration, is that in fermentation, the process does not require oxygen. It is an anaerobic process. Whereas if we talk about aerobic respiration, oxygen is very important. It is the final electron acceptor, which we have studied. So if we talk about the end product of fermentation, we generally have carbon dioxide or ethanol in case of alcoholic fermentation. And in case of lactic acid fermentation, we know it is lactic acid. So if we talk about aerobic respiration, our end products are water. So here in the image you can see, so there is incomplete oxidation of glucose first to produce pyruvic acid. This process of glycolysis, it is common to both fermentation and aerobic respiration. After this, in the presence of oxygen, aerobic respiration takes place, and in the absence of oxygen, fermentation takes place, which produces lactic acid or ethanol depending upon the fermentation, whether it is alcoholic fermentation or lactic acid fermentation. So here you can see in the diagram, now let's look at some more differences between them. Where does fermentation takes place? It takes place only in the cytoplasm. Aerobic respiration takes place in both cytoplasm and mitochondria. Because in case of fermentation, we know process of glycolysis takes place in cytoplasm. So the overall process takes place in cytoplasm. Aerobic respiration starts in the cytoplasm, that is glycolysis, followed by the products move into mitochondria, that is the mitochondrial matrix, as well as the inner mitochondrial membrane are involved. If we talk about the end products of fermentation, in case of alcoholic fermentation, end products are ethanol and carbon dioxide, as in the case of yeast. Whereas in case of lactic acid fermentation, the end product is lactic acid, that is in muscle cells. If we talk about aerobic respiration, what are the end products? Carbon dioxide and water. Now let's talk about the energy yield, that is the ATP. So the energy yield in case of fermentation is 2 ATP per glucose molecule. So it is very less. If we talk about aerobic respiration, the energy yield is 36 to 38 ATP per glucose molecule. So you can see the difference between the ATP yield of these two processes, showing us how important oxygen is. So again here in the diagram you can see, if we talk about the location, this process of glycolysis is taking place in the cytoplasm. In this process, 2 ATP is produced. This pyruvic acid, so it depends, different types of cells will handle this pyruvic acid depending upon the availability of oxygen. So if oxygen is present, the pyruvic acid enters the Krebs cycle, and then there is the electron transport chain, and finally we have production of water and ATP. And if we talk about fermentation, the process takes place in cytoplasm itself. Krebs cycle takes place in the mitochondrial matrix, and then the process takes place in inner mitochondrial membrane. If we look at the end products in this image, for fermentation, it is lactic acid and ethanol, depending upon the fermentation. And in case of aerobic respiration, if we look at the end products, it is carbon dioxide, water, as well as ATP. The ATP yield, if you look at in the diagram, there is only 2 ATP molecules produced which are coming from glycolysis. But if we talk about aerobic respiration, there are 36 to 38 ATP produced per glucose molecule after oxidative phosphorylation. Now let's talk about energy efficiency for both fermentation and aerobic respiration. We have seen that the ATP produced in case of fermentation is only 2 ATP molecules. So it is very less efficient in terms of energy. Whereas aerobic respiration is a much energy yielding process and therefore it is more efficient. Now let's talk about the final electron acceptor. So the final electron acceptor in fermentation is organic compounds like pyruvate or acetaldehyde. Whereas in aerobic respiration, we have seen that the final electron acceptor is oxygen, which produces water. In case of fermentation, there is incomplete oxidation of glucose, whereas in case of aerobic respiration, there is complete oxidation. If we talk about the steps involved, we have already seen that in fermentation, first step is glycolysis, and the second step is conversion of the end product of glycolysis, that is pyruvate, to its end product, either lactic acid or ethanol and carbon dioxide, depending upon whether it is lactic acid fermentation or alcoholic fermentation respectively. If we talk about aerobic respiration, the steps that are involved includes first glycolysis, which takes place in the cytoplasm, followed by link reaction, which is oxidative decarboxylation. Then there is Krebs cycle in the mitochondrial matrix, followed by the electron transport chain and oxidative phosphorylation, which takes place in inner mitochondrial membrane to produce ATP. So here you can see that starting from glycolysis, there is complete oxidation of glucose to the point till ATP is produced. Now let's look at some other differences. If we talk about NADH oxidation, NADH is converted back to NAD plus through substrate reduction in case of fermentation, whereas in case of aerobic respiration, this NADH is oxidized through the electron transport chain. If we talk about the duration, fermentation, it is a temporary process in most organisms, whereas aerobic respiration is a continuous process. If we look at some examples where these processes are taking place, fermentation takes place in muscle cells in animals during intense exercise. It also occurs when yeast, in case of yeast during brewing, and it also occurs in bacteria in yogurt production. If we talk about aerobic respiration, it takes place in most cells in plants, like majority of cells in plants. Aerobic respiration takes place in animal cells as well as many bacteria. Now let us look at some applications of these processes. Here also we can differentiate between them. Fermentation, it is useful in food production, that is for production of bread, wine or beer. And it is also useful in production of dairy products like yogurt and cheese. If we talk about aerobic respiration, our normal cellular metabolism depends upon it, and energy production in most organisms is aerobic respiration, and therefore it is very important. Environmental conditions, which means that fermentation occurs in anaerobic conditions, that is in the absence of oxygen, whereas aerobic respiration, it requires aerobic conditions, that is presence of oxygen is essential. So here you can see in the diagram, fermentation takes place without oxygen, whereas aerobic respiration takes place with oxygen. Students, now let's talk about amphibolic pathway. So amphibolic pathway, it refers to a metabolic pathway. So this amphibolic pathway, it can serve both as a catabolic, which means breaking down. So catabolism is the process of breakdown, and anabolic functions. Anabolic is building up, that is synthesis. So amphibolic means that there is both catabolic and anabolic functions. That is, there is both breakdown and building up taking place. So here in the image you can see, in anabolic reactions, smaller molecules build up to make larger molecules, and in this process, energy is utilized. Whereas if you look at catabolic reaction, which means breakdown, here larger molecules, they split, that is, they break down into smaller molecules with the release of energy. So let's first talk about catabolic reactions. So catabolic reactions, they break down complex molecules into simpler ones. And in this process, energy is released, and therefore it is known as exergonic. So exergonic reactions are those in which energy is released. Examples include of catabolic reactions. If you look at examples, it includes glycolysis and beta oxidation. So this process, because energy is released, and therefore it produces ATP, NADH and FADH2. And these products are used for energy or building blocks. That is, this energy that these products contain is used for building other compounds. Now let's talk about anabolic reactions. So what are anabolic reactions? They build complex molecules from simple ones. That is, smaller molecules are making up larger molecules with the input of energy. So in this process, because energy has to be given from outside, the process requires energy and therefore it is known as endogonal. Example of such processes includes gluconeogenesis as well as fatty acid synthesis. Anabolic reactions, they utilize ATP and NADPH because this process requires energy for building up larger molecules from simpler ones. And they create storage molecules and cellular components. So basically, they're utilizing the energy to create various storage molecules and other cellular components. Examples of such processes include citric acid cycle, that is the TCA, or that is the tricarboxylic acid cycle or Krebs cycle. Now, the citric acid cycle, it is a primary amphibolic pathway in cellular metabolism. What happens here? There is both energy production as well as biosynthesis taking place in TCA. Now let's talk about integration of different substrates. So in case of catabolic reactions, where do carbohydrates enter? So they enter at the glycolysis stage. Fats, if we talk about, because carbohydrates, we have seen that in glycolysis there is incomplete oxidation of glucose. So it begins with glucose. So here the substrate is glucose. If we talk about fats, they are first broken into glycerol or fatty acids. So if it is glycerol enters glycolysis as phosphoglyceraldehyde, whereas fatty acids, they are first broken down into acetyl coenzyme A and then they enter the Krebs cycle. If we talk about proteins are first broken down into amino acids. Amino acids are the building blocks of proteins. And these amino acids, after deamination, they can enter the pathway at various points. For example, they can enter as pyruvic acid, they can enter at the acetyl coenzyme A level, or a Krebs cycle intermediate. So, different substrates, they can enter the pathway at different levels. So, here we can see in the image that in case of fats, they are first broken down to fatty acids and glycerol. So, if fatty acid is to enter the pathway, it can enter as acetyl coenzyme A. And for glycerol, this is first converted to glyceraldehyde 3-phosphate or dihydroxyacetone phosphate. And then it further enters the cycle. If we talk about carbohydrates, which we have already studied, they are broken down into simple sugars like glucose. This glucose is then converted to glucose 6-phosphate, then to fructose 1,6-bisphosphate, and finally it enters through glycolysis, and then further there is the TCA cycle and other processes. Now if we talk about proteins, they are first broken down into amino acids, which after deamination enter the pathway at various points. So they can enter as here you can see as pyruvic acid or acetyl coenzyme A, or as a Krebs cycle intermediate. If we talk about anabolic reactions, intermediates from the pathway, they can be withdrawn to make other molecules. So these are processes where intermediates from metabolic pathways, they are used to create other essential molecules. And this is very important for plant respiration and metabolism. So acetyl coenzyme A, it is a precursor for fatty acids and steroids. So if we talk about alpha ketogluteric acid from Krebs cycle, it is a precursor for amino acid synthesis. So you can see that intermediates, various intermediates from this pathway, they are used to make other molecules, like we have seen for acetyl coenzyme A and alpha ketogluteric acid. All of these are the starting molecules for synthesis of other compounds. Students, now let's talk about the respiratory quotient. What is the respiratory quotient? It is the ratio of the volume of carbon dioxide evolved to the volume of oxygen consumed in respiration. Let's look at the formula. So what is RQ? It is the volume of carbon dioxide evolved during respiration divided by the volume of oxygen consumed during respiration. What does this RQ value indicate? So it tells us the type of substrate that is being used in respiration. So we can calculate RQ for various compounds. First, let's calculate for fatty acid, that is tripalmitin. So here in the reaction, you can see tripalmitin. In this process, oxygen is consumed, carbon dioxide is released along with water and energy. So here you need to note that 145 oxygen molecules are being consumed and 102 carbon dioxide molecules are being evolved. So what will be the respiratory quotient? It will be volume of carbon dioxide evolved by volume of oxygen consumed. So from the reaction, we can get the values 102 carbon dioxide divided by 145 oxygen, which gives us a value of 0.7. Now let us look at the RQ values for different substrates. So various substrates are given in the table. So let's start with carbohydrates. So the respiratory quotient for carbohydrates is 1. What is the reason? Because here equal amount of carbon dioxide and oxygen are evolved and consumed. So the ratio gives us a value of 1. If we talk about fats, the RQ value is less than 1. For example, in case of tripalmitin, we have seen that the ratio was 0.7. Because fats are rich in hydrogen, so they require more oxygen for complete oxidation. So because more oxygen is being consumed than the carbon dioxide that is being evolved, the ratio is less than 1, which gives us 0.7 in case of tripalmitin. If we talk about proteins, the ratio is almost 0.9. It is an intermediate value between carbohydrates and fats. And if we talk about organic acids, the respiratory quotient is more than 1. Why? Because they are rich in oxygen. Now let's look at a special case, which is an exception. For anaerobic respiration, carbon dioxide is evolved, but no oxygen is consumed, because we know in a case of anaerobic respiration, there is no oxygen consumed, there is no oxygen available in the first place. So only carbon dioxide is evolved, and therefore here the ratio or the respiratory quotient is infinite, because our denominator is almost 0. So students, now let's take a small recap of what we have learned so far. We have talked about how the respiratory balance sheet shows oxygen consumption and carbon dioxide production during cellular respiration. Amphibolic pathways, that is those which involve both catabolism and anabolic reactions, like the TCA, that is the tricarboxylic acid cycle. They serve both catabolic and anabolic functions. So there is breakdown of molecules, larger molecules, into simpler ones, as well as synthesis of larger molecules from simpler ones. And that is what we call amphibolic pathways. Then we also studied about the respiratory quotient. What is it? It is a ratio of carbon dioxide produced or evolved to oxygen consumed, indicating the type of substrate which is being used during respiration or the type of substrate that is being metabolized. We saw for carbohydrates, respiratory quotient is 1. For proteins, we have seen that it is nearly 0.7 or 0.8. Now let's solve some NCERT based questions. So the first question is, what is the respiratory balance sheet and why is it important in metabolism? So we studied that the respiratory balance sheet, it is a comparative analysis of carbon dioxide production and oxygen consumption that takes place during cellular respiration. Now, why is this respiratory balance sheet important? Because it helps us calculate the efficiency of metabolic processes. Like it tells us the energy production. So that tells us about the energy efficiency of the metabolic processes involved. Then it also provides information about the type of substrate that is being used during respiration. That is the type of substrate that is being oxidized. It also helps us determine the respiratory quotient, which is the ratio of carbon dioxide evolved to the oxygen consumed. And then it assists in understanding the overall energy metabolism of organisms. Further, this respiratory balance sheet is also useful in clinical settings for metabolic assessment. Now let's look at the next question. How does the respiratory quotient value help identify the type of substrate being metabolized? Let's look at the answer. So the respiratory quotient value, what it indicates? It indicates different substrates. Now how? For carbohydrates, if we talk about pure carbohydrate oxidation, the RQ value is 1 because equal volume of carbon dioxide is evolved and equal volume of oxygen is consumed. So the ratio is 1. If we talk about fat oxidation, the RQ is 0.7. More oxygen is needed due to higher hydrogen content. If we talk about proteins, the RQ value is 0.8. So for protein oxidation, because it is an intermediate value due to the amino acid composition. Mixed substrate utilization gives intermediate values of RQ. That is, if not one particular type of substrate is being used, then we get intermediate values of respiratory quotient. And values more than 1 indicate carbohydrate conversion to fat. Now let us look at some keynotes. So we have studied that respiratory balance sheet, it shows us a net gain of 38 ATP molecules during complete oxidation of glucose. This is important. We have talked about amphibolic pathways which include both anabolism and catabolism. That is TCA cycle, it acts, and the example that we have seen is the TCA cycle, which acts as both catabolic, that is there is breakdown of larger molecules into simpler ones with the release of energy, and anabolic pathway, that is synthesis of larger molecules from simpler ones with the input of energy. Okay. Then we studied about the respiratory quotient. This is also very important. It is the ratio of volume of carbon dioxide evolved to the volume of oxygen consumed. And the respiratory quotient for carbohydrates we calculated came out to be 1. For proteins, it is approximately 0.9. For fats, it is less than 1, that is approximately 0.7. Now let us look at some interesting questions which are for you to solve. So think about them. First question asks, how does the respiratory quotient of protein differ from that of carbohydrates? So we know for protein it is almost 0.8 and for carbohydrates it's 1. So you can compare the ratios. to see how much carbon dioxide and oxygen are involved. The second question is, how does the respiratory question change during the transition from a fed to a fasted state? That is, in a scenario where a person has eaten and another scenario where there is fasting, that is no food intake. So you have to compare the respiratory question change for both scenarios. Then calculate and explain why respiratory quotient for palm oil, primarily composed of palmitic acid, would be different from that of glucose oxidation. Next question. How does the respiratory balance sheet help in understanding metabolic disorders? So students, now let us solve daily practice questions for today's class. So the first question is, which of the following is the common pathway in both aerobic and anaerobic respiration? The options are electron transport system, Krebs cycle, glycolysis, or glycogenolysis. So we have to tell which pathway it occurs in case of both aerobic as well as anaerobic respiration. So let us look at the answer. So the answer is glycolysis. So we know glycolysis, it takes place in the cytoplasm. In case of both aerobic and anaerobic respiration, it is common to both, where there is incomplete oxidation of glucose to produce pyruvic acid. Later, this pyruvic acid, it is handled by different cells depending upon oxygen availability. In case of anaerobic respiration, there is either alcoholic fermentation or lactic acid fermentation, which converts the pyruvate to ethanol, carbon dioxide, or lactic acid. And in case of aerobic respiration, we know there is presence of oxygen. So there is then complete oxidation of that glucose to produce water, carbon dioxide, and ATP. Now let us look at the next question. Which assumption is necessary for calculating the theoretical net gain of 38 ATP per glucose molecule in aerobic respiration? Let us look at the options. Intermediates are used to synthesize other compounds. Option 2. All pathways operate simultaneously. Third option. NADH from glycolysis is transferred to mitochondria. Or the fourth option. Alternative substrates enter at intermediary stages. So let's look at the answer. So the assumption that is necessary is that NADH from glycolysis is transferred to mitochondria for calculating the theoretical net gain of 38 ATP per glucose molecule. However, these assumptions are not very true in case of real cellular processes. Let us look at the next question. What is the key difference between fermentation and aerobic respiration? The options are: fermentation uses the TCA cycle. Aerobic respiration completely oxidizes glucose to carbon dioxide and water. Fermentation generates more ATP per glucose than aerobic respiration. Or the fourth option, aerobic respiration does not involve NAD plus generation. So you have to differentiate between fermentation and aerobic respiration. Let's look at the answer. So the major difference between fermentation and aerobic respiration is that aerobic respiration completely oxidizes glucose to produce carbon dioxide and water. Whereas if we look at fermentation, there is not complete oxidation of glucose. That pyruvic acid, it undergoes alcoholic fermentation or lactic acid fermentation. In alcoholic fermentation, we have studied carbon dioxide and ethanol are produced. Whereas in case of lactic acid fermentation, lactic acid is produced. So there is no complete oxidation of glucose. Whereas in case of aerobic respiration, there is complete oxidation. If we look at the other options, Fermentation does not involve the tricarboxylic acid cycle. This is wrong. Fermentation generates more ATP per glucose. This is also incorrect. Only 2 ATP molecules are produced. Now let's look at the next question. Which factor contributes to higher ATP yield in aerobic respiration compared to fermentation? Option A, absence of glycolysis. Option B, utilization of electron transport chain. Option C, production of ethanol as an end product. And option D, occurrence in the cytoplasm. So let's look at the answer. So the factor which contributes to this higher ATP yield in aerobic respiration is the utilization of electron transport chain. Because here the proton gradient is generated, which drives the ATP synthesis process, and therefore this is the factor which is responsible for higher ATP yield. Because in case of fermentation, the only energy that is produced is the 2 ATP molecules from glycolysis. However, in case of aerobic respiration, there is electron transport chain where breakdown of a proton gradient results in synthesis of ATP. Let's look at the next question. Why can't the theoretical 38 ATP per glucose be achieved in actual cellular conditions? First option, all pathways function independently. Second, intermediates, they are never used for other synthesis. Third, mitochondrial NADH transport is 100% efficient. Or fourth, assumptions like sequential pathway operation are invalid. So let's look at the answer. So why isn't the theoretical 38 ATP per glucose molecule be achieved? Because assumptions like sequential pathway operation are invalid. Because in case of actual cellular conditions, mostly the pathways, they are simultaneous rather than sequential. So this was all for today's class.